Hi guys. So today in this video, I'm going to go over cellular respiration. And now cellular respiration is the inverse process from photosynthesis. In this process, we are taking a sugar molecule, glucose, and we are breaking it down, breaking those bonds apart to release the energy that was stored in those bonds. And so the general equation for this storyline is glucose and oxygen as reactants with water and carbon dioxide as our products. Now, this particular reaction is for aerobic respiration. Aerobic respiration is respiration that utilizes oxygen. There is cellular respiration that does not utilize oxygen, and that's called anaerobic respiration. So when we take this sugar and we reduce it down to simpler molecules, we are increasing the amount of entropy in this individual instance. And when we increase the amount of entropy in an instance, we release energy to do work. And so by breaking this complex ordered glucose down into these smaller, less ordered molecules, we are creating an entropically favorable situation where we get a little bit of free energy to do something with. Now, aerobic respiration is a lot more efficient than anaerobic respiration. So we as complex multicellular organisms cannot persist on just anaerobic. We need to use aerobic. Smaller things like bacteria can be anaerobic. Our cells, in instances where we don't have access to enough oxygen, can perform anaerobic respiration. There are some side effects, such as the buildup of lactic acid, that are detrimental, and the production of ATP is too low to sustain those cells for very long. Right? But it's better than nothing. So an overview of our general process here. Right? Our cellular respiration, our aerobic cellular respiration, occurs in three major steps. The first step is glycolysis, the second step is the Krebs cycle, and the third step is oxidative phosphorylation and chemiosmosis via an electron transport chain. Yeah. You can shorten it to either oxidative phosphorylation or electron transport chain. Either one is an effective way of demonstrating what stage you're on. Okay. So glycolysis, way back here, happens in the cytoplasm of the cell. All cells have cytoplasm. All cells can do glycolysis. Right? This is an anaerobic step. Yeah. And so glycolysis, glyco referring to sugar, lysis referring to splitting, effectively takes a glucose and rips it in half. It's more complex than that, but that's basically the outcome. Those two halves are pyruvates, which is to say another name for pyruvic acid. So they are molecules that are 3 carbons long. In that process, I also get an NADH produced, right? And so this is an electron carrying molecule. It can move high energy electrons from one place to another, right? Similar to NADPH from photosynthesis. The pyruvic acid, the pyruvates, are shuttled into the mitochondria, right? Where first, almost immediately, they are converted into an acetyl coenzyme A. It's just a different molecule. Okay? It's a 2-carbon molecule. That happens in this region of the membrane of the mitochondria, right? So it's between the outer membrane and the inner membrane of the mitochondria. I get my pyruvic acids converted into acetyl coenzyme A's. Okay. They then enter the Krebs cycle, which is also happening in that same location. The Krebs cycle is a series of reactions whose main purpose is to generate NADH and FADH2, both high energy electron carrying molecules. It also produces CO2 as a byproduct. These electron carrying molecules are then shifted to the inside of the interior membrane of the mitochondria, where they are utilized to complete oxidative phosphorylation, and they generate ATP, which is the whole point of doing this process, is to take the energy out of sugar and to move it into ATPs that the cells can use for work. Water is a byproduct of oxidative phosphorylation. Okay. So, moving into a little bit more detail. Okay. Step 1, I'm starting with my glucose, this bad mamma jamma right here. Okay? I'm going to slap it with some ATP. I'm going to slap it with some small little carbon groups. Okay? And I'm going to turn it into this symmetrical looking guy right here. Okay? So I replaced this alcohol group with this carbon and phosphate group, and I've phosphorylated this side over here. Okay? So the phosphates came from the ATP. My small carbon group was used to replace my alcohol group. So I got this symmetrical guy. Cool. All right. I then slap it with an enzyme. There are some intermediary steps that I've not included here because they're beyond the scope of this course, but effectively, after getting slapped with this enzyme, we get 2 G3Ps. And if you recall from photosynthesis, G3Ps were our precursor molecule to our glucose. Right, so we're basically doing the reverse of photosynthesis here. I've taken my glucose and I've split it into 2 G3Ps. I went through pyruvic acid to get there, but same end game. Right? Okay. So I got my 2 G3Ps, and then I slap them with some more enzymes. I said something I don't like. Rewind about 30 seconds. Okay. All right. So I got my 2 G3Ps, okay, and we're using them to make pyruvic acids. Okay, cool, awesome. So I got my G3Ps. I've converted them with some enzymes into pyruvic acid. That process also generated for me an NADH and some ATPs. So we are looking here at glycolysis, which, as I said earlier, is an anaerobic step. Right? And so now we have to ask ourselves, are we moving into aerobic respiration, or are we staying anaerobic? So if we're moving into aerobic respiration, my pyruvic acids were the products. That was the point of doing glycolysis, right, to get this stuff. I pass that into the mitochondria and move on to step 2. If I'm doing anaerobic respiration, okay, my ATP was the product. That was the point of doing glycolysis, was to get these measly 2 little stupid ATPs. Right? And my pyruvate and my NADH are trash. Right? So in that case, if I'm doing anaerobic, I'm going to recycle pyruvate and recycle NADH so that I can start glycolysis all over again. Okay? The process that recycles those 2 molecules is fermentation. Okay? And so anaerobic organisms will do fermentation to regenerate the starting products so it can do glycolysis to make tiny little ATPs. Okay? We're ignoring that for now. We're going on to step 2. We're doing aerobic respiration. All right. So step 2, broken down into 2A, which is before the Krebs cycle. Right? So this is where my pyruvic acid plus my coenzyme A become acetyl coenzyme A. I lose a CO2 when I rip off one of my carbons by CO2, okay? And I get an NADH through this process, passing to step 3. But here's my product, acetyl coenzyme A. It's a 2-carbon molecule, and this is going into the Krebs cycle, which is step 2B. Okay. So my Krebs cycle is taking acetyl coenzyme A, which I just made, and another molecule, oxaloacetate. Okay. So my acetyl coenzyme A is a 2-carbon molecule. My oxaloacetate is a 4-carbon molecule. I slap them together to get a 6-carbon molecule. Okay. So this is basically the starting point of my Krebs cycle. Right. Then as soon as I have that 6-carbon molecule, I'm going to rip one of those carbons off. Just one. Plop it off. A little bit of energy comes out, and I use that little bit of energy to generate an NADH. Okay. And then CO2 leaves as trash. And so the carbon that I plopped off is going as CO2. Then, when I have my 5-carbon molecule, I'm going to rip another one off. Okay? I use the energy that's released from that ripping to create another NADH and another trash carbon dioxide. So now I'm down to a 4-carbon molecule. It still has that coenzyme A group attached to it. We're going to rip that off. All right, back again. So, where was I? I think I was on the 4-carbon molecule loses its coenzyme A. Okay. And when that coenzyme A pops off, we use that energy to make an ATP. Okay. So now I'm down to a 4-carbon molecule without my coenzyme A. It's not exactly super happy in the format that it is, and so it's willing to give up a couple of its hydrogens. Those hydrogens are used to generate FADH2. And now I'm down to a different 4-carbon molecule, but still a 4-carbon molecule. And I slap that 4-carbon molecule with some water, okay? And that is used to generate some NADH, okay? And convert my 4-carbon molecule back to my oxaloacetate, which is the starting molecule, right? So this brings us back to step 1. Very important. Because otherwise it would stop working, right? That's what makes this a cycle. Cool? So in this whole story, I've made 3 NADHs and an FADH2 and an ATP. Now, that's with each pyruvic acid. So for each glucose, I get to do this twice. The NADH and the FADH2 are passed to step 3, which is oxidative phosphorylation, using the electron transport chain and chemiosmosis. And that's embedded in the interior membrane here, okay, of the mitochondria. So in the mitochondria, we just came from the Krebs cycle. And so this membrane is representing this membrane. This is the green region, so that's on this level in the sandwich part. This is the blue region, so this is on the very inside of the mitochondria. Okay. So I have some protein complexes embedded in this interior membrane. These protein complexes are the electron transport chain because it transports an electron, electrons actually. So the NADHs and the FADH2s are going to get involved. Okay? They're going to give up an electron. It's a high energy electron. That electron is going to make its way into the electron transport chain. The H is going to fall off as an H+. NAD+ is going to leave, go back to be recycled. But now this high energy electron is passed from protein to protein in the electron transport chain. As it does so, the proteins are using the energy from the electron to pump the hydrogen ions out. So from the inside to the sandwich zone. Okay? And so I'm building up a concentration of H pluses in the sandwich zone. Okay? So I got one H plus pumped here, I got some H pluses pumped here. Okay, the electron gets passed. Okay. Eventually the electron is now a lower energy state and it needs to be dumped off and find a home. So that electron gets dumped off into an oxygen and some hydrogens, and it leaves as water. So the whole reason that we need to breathe in oxygen is to get this oxygen right here, right? So we have a place to dump this electron onto. Okay? So this is the oxygen in and the water out part of the storyline for cellular respiration. The whole point of that was to generate this concentration gradient of H pluses in this sandwich zone. So the H plus is high concentration here, low concentration here. Rules of diffusion say they want to move down, right? But they're ions, so they can't just cross the membrane. They need a channel. The only channel to go back in is this protein right here. This is ATP synthase. It's an enzyme that makes ATP. It's a really cool enzyme. Right? And so as the H pluses move down the concentration gradient, right, they possess a kind of potential energy. And as they move through the protein structure, this enzyme right here, that potential energy is converted into a kinetic energy via a spinning mechanism. Right? This little dude actually spins. Right? So he has a rod on the inside, he has a rod on the outside, and this bottom part rotates around that rod. Okay? So the hydrogen comes down like a little spiral slide type of thing, spins the bottom of this enzyme, and that takes the 2 active sites, one of which is engaged with an ADP, one of which is engaged with a phosphate, and it smushes them together to make ATP. Boom. Magic. Right? Not magic. Science. Magic science. Yeah. And so that's how I make my ATPs out of this whole storyline. Boom. My ATPs here. So every glucose that I break down nets around 30 ATP through this process. And the gradual removal and addition of different things along the way helps control the release of energy. If you were just to explode the glucose in one fell swoop, the energy would come out. A lot of it would come out as heat, it would come out as light, other wasteful things that we don't want to do. Right? We want to carefully harness each little piece of energy and use it properly. Right? And so by doing it step by step by step through all these convoluted processes, we are able to more efficiently harness that energy and make sure that more of it ends up in the bond of ATP. Right? So ADP, you slap together that extra P on the end, and you get yourself some energy stored in that tiny little bond. Right? So in this case, I decreased entropy just a little bit. Right? And so I stored a little bit of energy in here. And when I break that P off, I increase my entropy again, and I release a little bit of energy. Okay? So this is simple enough that the cells can actually utilize it to do work. Right? It can phosphorylate different things. It can change the structure of different things. And by changing structure, you change function, you make things happen. Okay? So cellular respiration takes sugar, glucose, runs it through glycolysis to break it into pyruvates. Those pyruvates are sent into the mitochondria. They go through the Krebs cycle to slowly remove carbons to harness energy. We store that energy in the electron carrying molecules, NADH and FADH2, which get passed into the interior section of the membrane where the electron transport chain can occur, where we use the energy in those high energy electrons within the electron carrying molecules to pump hydrogen ions into the sandwich zone. That's not the official name, that's what I call it, the sandwich zone. Right? And I have a high concentration of H+, gives me potential energy. As they come down that concentration gradient, it is converted into kinetic energy. We use that kinetic energy to create ATP. And that's the end game. It's beautiful. It's wonderful. It's lovely. I love some cellular respiration. All right, that's it. Bye.